All right, welcome back. We are going to go through the first step of cellular respiration, which is glycolysis. Obviously, being the first step, it's actually a very ancient step and really important to the evolution of life. So again, what is the whole point of all this? Is to get ATP. Cells need energy to do all those things we talked about, to move, to cause chemical reactions to happen. So how do we get ATP? Well, number one, what does glucose mean? We break the word apart. Glycolysis, glyco meaning sugar, and lysis meaning breaking. So glycolysis to break sugar. So let's recall that glucose is normally in a ring shape, but it's a 6-carbon molecule. There's lots of OHs and Hs here, but those aren't really that important. Because remember, the bonds between carbons is where a lot of the energy of sugar is stored. So the first step is we take 6 carbons and we break it into 2 3-carbon molecules. So we break it in half, and this bond right here is the first bond broken in breaking down sugar. However, it's actually really inefficient. We only generate 2 ATP net for every 1 glucose. So remember, net means overall. You actually gain 4, but you have to spend 2 to get it, and we'll talk about that. You do need to know that it occurs in the cytosol or cytoplasm. So glycolysis happens not in the mitochondria. I know the mitochondria is the powerhouse of the cell. It does provide lots of energy, but glycolysis happens in the cytoplasm. Remember, ancient pathway. There was no mitochondria in every cell at the beginning. So what happened? Well, to start off, why is glycolysis an anaerobic process? Well, because there was no oxygen early on. We talked about that in the origin of life. All the oxygen that existed at the time reacted really quickly, so there was no free O2 floating around. And the first cells had no organelles. They didn't have any mitochondria to make a lot of energy. So prokaryotes was the original glycolysis producers, and that pathway has been passed down through all species. It's pretty much in almost all cells use glycolysis, which is actually another good piece of evidence for evolution too. So what we need to know is that prokaryotes were the first to develop this glycolysis, the first step in breaking down sugar and getting energy. All right, so this diagram looks really intimidating. We'll kind of break it down step by step. But the big idea is that we take glucose, we add ATP that reacts with the glucose. So the first step is ATP is going to become ADP. Ooh, that means it lost a phosphate. Well, what happened to it? It actually loses the phosphate it binds to each half of the glucose. So this is each called a pyruvate molecule. Now, the phosphate, remember, has lots of energy. That's a very energetic functional group. And so by adding this phosphate, we're actually going to add another phosphate and in the process make this NADH. Ooh, NADH, that's something new. We need to remember that NAD is the less energetic and NADH is a more energetic molecule. We now have these very energetic double phosphate pyruvate, and that's actually going to remake 2 ATP. And since we broke it in half, this is going to happen twice. All right, and so in the end, we're going to make 2 NADH and 4 ATP. But remember, we actually used 2 to do it. So let's break this down step by step. Glycolysis is endergonic. It requires some ATP in, 2 ATP specifically. But we get 4 ATP out and 2 NADH. Think of NADH as like a little piggy bank. We're going to take some electrons with us that have a lot of energy, and we're going to use them later in the process. Okay. So one little thing I want you to add to your fundamental sheet is that more letters equals more energy. That's kind of a good way to remember this. So NADH versus NAD plus. Ooh, NADH has more letters. That means bigger, better, more energy. Sounds good to me. All right, so let's go through those reactions real quick. The first half of glycolysis is called priming. We're going to add a phosphate to each of the glucose. You don't need to know this step like super well, just kind of the idea that we're going to use ATP to make this more energetic. This makes it really unstable. The second half is we're going to then harvest all the energy that's in the sugar. We're going to make NADH, again, that piggy bank molecule. So NAD plus turns into NADH. We got more letters. and then ATP is going to be produced. We're going to turn the glucose into pyruvates, and then those phosphates go back to the ADPs to make ATPs. Don't worry too much about substrate level phosphorylation, but this is just the idea that we're going to actually add phosphates via our substrate. So adding the phosphates to make ATPs with our reactants. So again, the idea we really need to know is what goes into this process and what comes out. We're adding in 1 glucose and 2 ATP, and ultimately we end up with 2 NADH, 4 ATP, and 2 3-carbon sugars we're going to call pyruvic acid. All right, if you get that, you're good. You don't need to understand all this nitty-gritty chemical business. So this last step, there's actually an enzyme that helps with this making of ATP. So this is called a kinase. A kinase is a really broad category of enzymes. Kinases change phosphate groups. So they normally add a phosphate to something. So you can see in our picture, this kinase is going to move the phosphate over to ADP, and then boom, we get ATP out of it. So the phosphate directly transfers over phosphate groups from the fuel, this pyruvate, to make ATP. So that's what substrate level phosphorylation really is. We're going to add a phosphate to our ADP. So let's do our math. We used 2 ATP. We got 4 ATP out. So really, you know, you only with your investment, you only get 2 ATP in the end. That really seems like a whole lot of work. And so let's go ahead and add our glycolysis reaction to our really important fundamental list. It's a pretty big reaction to know. So for what goes in number 4 is glucose, 2 ATP, and 2 NAD pluses, because those get turned into NADHs, which we don't really know a whole lot about. Again, that's kind of something we'll get to on Friday. And then out comes these 3-carbon pyruvates, are 4 ATP, so remember 2 net ATP, and that is also making 2 NADH piggy banks. Remember, this is happening in the cytosol, so feel free to pause and copy that down. But we can't just stop there, okay? Remember, everything's a cycle. If we use up our ATP and we use up our pyruvate, we got to make more of this. And we can't just keep making NAD+. That's going to take too much energy. So we have to make sure we can turn this NADH back into NAD+. It's a limiting reactant. All right. So something has to turn this NADH back into NAD+. We have to get rid of this H, which we'll get to. So what's going on? Is that all the energy that we're going to get? Well, for now, and for really old organisms, yeah. For a billion years, that's how everything got energy, is they used glycolysis. They didn't have oxygen. They couldn't react it that quickly or that well. And if you only relied on that reaction for sugar, you'd get about 3.5% of all the energy stored in sugar. That's not a whole lot. So you're going out eating all these things, and you're only getting 3.5% of your value. And so you can either go down this O2 route to remake your NAD+ and do the Krebs cycle and electron transport chain in your mitochondria. This is where you're going to get a lot closer to that 100% of the energy in sugar. This other route with no O2 is called fermentation. This is how prokaryotes and simple organisms remake that NAD+. So remember, we got to remake this NAD+ in order to keep this reaction going. So that is what these two processes do, is they help regenerate and use that NADH. Here's a quick little schematic. So you have glucose going into your cell, and glycolysis then happens in the cytoplasm, inside our cytosol. And so this pyruvate can go, again, in one of two directions. We can either go into fermentation and anaerobic respiration, no oxygen, or NO2, as I abbreviated it. Or if you have oxygen, then you're going to go in the mitochondria and you're going to do all this cool stuff we're going to talk about on Friday. So let's talk about this. What do like really ancient things do? Well, they do fermentation. So there are two types we're going to talk about. Remember, this is anaerobic, which means no oxygen is needed. And so this is a metabolic process which we're going to regenerate more of that NAD+ that we need. So we're going to create lactic acid as an issue. So what does this? Well, bacteria does this. You do this, oxygen-deprived muscle, and this will cause, muscle cramps in athletes. So why do we do this? Why don't we just always rely on mitochondria? Well, sometimes you need a lot of energy fast. Okay, so maybe you're in the forest and there's a bear, and the bear starts chasing you, and you're like, oh man, I gotta go. So you start running and running and running, and you're gonna start using up all your ATP, even faster than your mitochondria can make it. And you still got to keep running and get away from this bear, and you don't want to die. And so your body's like, fine, I will get you some ATP through any means necessary, and that is lactic acid fermentation. It's going to make a tiny bit of ATP really fast. You're not using all that sugar, remember, all that energy that's in it. So you're going to get away from the bear, cool, but then you're going to pay for it a little bit later. You're going to have really sore muscles. You got to deal with this lactic acid waste. Sprinters will experience this when they're in a race and they want to try and beat Usain Bolt. They're going to try and run really quick. So it gets a little bit of ATP really fast. You're not getting a lot. It's not efficient, but you're going to get some that you need in an emergency. How do we take advantage of this? Well, we use bacteria to ferment to make things like sauerkraut, cheese, soy sauce, and yogurt are all byproducts of bacteria creating this lactic acid, which some of us like in our food. That's number 9. All right, so what goes in that pyruvate? So the pyruvate goes in, and what comes out is you're going to turn that NADH back into NAD+. And so the two things that go in is pyruvate and NADH, but what comes out is NAD+. Cool, we can use that in glycolysis really fast, but you're generating a lot of this lactic acid, which can be bad for you. So glucose to pyruvate, cool, ATP, but we got to keep this NAD+ cycling. And that is what lactic acid fermentation does. And so what happens with all that lactic acid? Like, why aren't you sore forever? Well, you can reverse it. So you can turn the lactic acid back into pyruvate, but only when there's oxygen available. So just to summarize, 2 pyruvate go in along with 2 NADH, and we can remake that 2 NAD+, but you're generating 2 lactate as a byproduct. All right, so yeast do something different. They do alcoholic fermentation. This is not something humans do. So if you go out and run really hard, you're not going to get really drunk. Okay, we do not generate alcohol as a byproduct like yeast do. But this is their system for getting back NAD+. Instead of creating lactic acid, their byproducts are carbon dioxide and ethanol. Remember, this is anaerobic, no O2, very ancient. So yeast do this, and maybe some bacteria. And who takes advantage of this? Humans. We can make alcoholic beverages. So we stick yeast in bottles with no oxygen and say, hey, go at it. Turn all this sugar into alcohol. And actually what happens is the yeast keep doing that so much that they generate a high enough concentration of alcohol, around 12%, that they actually die in their own alcohol. And so if you want any higher percentage, you have to do distilling, like we talked about with moonshine and making distilled alcohol. But we also use it in bread. So why bread gets all fluffy is the CO2 that's generated actually makes the bread puff up. And most of the alcohol just evaporates. And so your bread normally doesn't have much in that. So what goes in? The pyruvate goes in along with the NADH, so just like lactic acid fermentation. But what comes out is more NAD+, the O means out, ethanol, which is alcohol, and then a CO2. And so this is like bacteria and yeast, whereas animals and some fungi and bacteria do lactic acid and turn NADH back into NAD+, using the lactic acid method. So make sure you know that both of these are anaerobic, no O2, and that different organisms have different processes, but the goal is the same. And so this relates to your yeast lab. So if you're thinking about the questions on the yeast lab, be thinking about what's going on when I put the sugar and yeast in the containers that you did the lab with, and that'll help you answer some of the post-lab questions. And so lastly, I talked about how eventually around 12% ethanol, the ethanol kills the yeast, and you can't reverse this. You can't turn the ethanol back into pyruvate. So again, it's a very ancient process and one that is just necessary for the early organisms to survive. So again, why do organisms do that? Like, why don't they evolve and get better? Why are they just relying on glycolysis and fermentation? Well, those organisms are really small. I mean, if you're a tiny yeast cell or bacteria, 2 ATP might be a lot. Cool, I can do the 7 characteristics of life. Fermentation works for me. And,